We are going to deal with mostly with bacteria, and we will give a little reference to archaea as we go along to see if there are any differences, major differences. We will talk a little bit about the microscopic eukarya also, but not too much. We do want to make it into the bacterial and the archaeal groups only. Okay, so with that, let's go ahead and see what you will be learning through this chapter. When you finish studying this chapter, you will be able to know what is a prokaryotic cell and how is it different from a eukaryotic cell. What is the prokaryotic cell composed of? What are different nutritional types of the prokaryotic cell? And what is this classification that is based on? The structures that are typically prokaryotic cell, what are functions of these structures, and major differences, structural differences between different types of bacteria such as gram-positive, gram-negative, acid-fast, mycoplasma, chlamydia, cyanobacteria. These are major groups of bacteria that we will refer to and how are the archaeal cells different from bacteria. So by the end of this chapter, you should be able to answer all these questions. So we first look at the slide that I had shown you when we started this semester with this class, that the groups of living forms, they have been put together in a phylogenetic tree, as we call them, and this is a phylogenetic relationships in all these life forms, and this has been based on sequencing the ribosomal RNA sequences. And here you see the branching off of these different groups, and you can see that bacteria and eukarya have a common ancestor here, whilst the archaea and eukarya have a common ancestor here. So by looking at this phylogenetic tree, you can see some relationships to each other and how far apart they are in their similarities or in their DNA structures. So it gives you a little bit of an idea that even though we'll be capitalizing and working mostly on this bacteria group, archaea seems to be between the two, and there are some similarities of archaea to bacteria, and then there are some similarities of archaea to the eukarya also. So just to give you a little idea, we are using a lot of comparative genomics these days, where you set of analysis by which gene function and evolution can be inferred. When you study the similar nucleotide and amino acid sequences in different organisms, you come up with this comparative genomics. So definitely this comparison within the domain will reveal some information about the genomic structure and evolution also. And in bacteria, we also have what we call horizontal gene transfer. And this horizontal gene transfer also changes the genome structures. And therefore, we can, by looking at the genome structures, you can decipher if there has been a horizontal gene transfer also. So when we are looking at all this information, the genomics, it tells us that these microbes have evolved and they have evolved into different ecological niches. And this becomes an important information piece for us in developing new therapeutic agents to say whether such a therapeutic agent will be able to control this type of a microbe or not. And to give you a little bit of just to tell you how we have looked at these comparative genomics by looking at the genome sizes of all these different names. So I would like you to focus on some of the names that you are familiar in the bacteria group that we always studied a lot as our model microbes, such as your Bacillus subtilis here. And then, of course, there is your Pseudomonas aeruginosa here. And then, of course, there is your E. coli, Escherichia coli K12 versus the regular E. coli. So they are E. coli K12 is here and E. coli 0157H7 is here. So the genomic sizes have been looked at. And what does ORF stand for? Anybody is familiar with ORF?
Open reading frame.
Open reading frames. And what do open reading frames signify?
Transcription, translation.
Meaning that there are, these are genes that can be transcribed and translated. So reading the whole genomics, of course, tells you. It's kind of interesting to see that the mycoplasma, which is the smallest of the bacteria group that we have in terms of size also, is also a one that carries a very small genome. And the ones that we in this group, as we look at it, chlamydia, rickettsia, these are very special ones. Rickettsia is again a parasitic. It has to have a live cell to be able to replicate. So you can see that those that are in the bacteria group that are in the lowest of this chart, they definitely have very small genomes, and they actually need help from other living forms to be able to replicate. So they are almost closer to, as we would say, viruses as such. Looking at your archaea group, you can see that they go from, if you're looking at the genome size in their sizes, there are the small ones here, which is almost the same as a bacterial small size, and then we have the large ones, which are huge, but we can see that some of the bacteria have bigger genomes also. Just to give you an idea as to how much work has been done in terms of comparative genomics, that we know the gene sequences of quite a few unicellular microbes that we work with in microbiology. Here are some of the clades of bacteria and archaea that you may be familiar with also, because we will sometimes talk about these different groups. We know we have always worked with proteobacteria. The proteobacteria group are the ones that are divided into beta, gamma, alpha, epsilon, and delta groups, from which you have some very well-known microbes that have been studied. Whilst we have the ones, the gram-positive bacteria are a little farther away. If you're looking at the tree here, the phylogenetic tree, you can see them branching off in different ways. So here is the archaea group, and here is all the bacteria that have been put into different clades as we go on and divide them. Just to give you an idea how diverse these bacteria are. Now, generally, when you've been studying microbiology up to now and bacteria, you've always focused on some very typical examples of bacteria, and you formed a idea that all the bacteria will look the same and function the same way. But we're going to take you away from your standard, structures and all, and we're going to definitely look at some very diverse group of microbes that you have never studied, and we'll see how different they are, not only in their structure but in their physiology, in terms of what are the capabilities, what the nutritional types will be different, and their functioning will be very different also. So just to give you a brief idea as to how diverse this group of bacteria are that we're going to be studying. Here is another major subdivisions in the prokaryote. This is only for references. Those things are just to give you the idea that how closely related they are and how far away they are. So here again, you will see your common names that you're familiar with. Your enterics are here in your special group, alpha, beta, gamma. So these are the gamma ones. This is the Escherichia, Klebsiella, Salmonella, Serratia. These names are coming back to you, I hope. We will be talking more about the Thiobacillus. We will be talking about the Caulobacter and Desulfovibrio, and those different groups that you have never studied. We will be looking at those also in this course now. And here are some of the subdivisions of the archaea. We will refer to some of them, especially the Halobacterium, the Methanococcus, and all the others that we will look at, Thermoplasma, and so on. Okay, so we'll just kind of deal with the archaea really quickly and put some pointers for you to say how different they are. So as such, archaea have many features in common with the eukarya, especially their gene coding proteins, encoded proteins. Their replication, transcription, and translation are very similar in terms of being similar to the eukarya. But there are features that are common with bacteria. Their genes for metabolism have some commonality with the bacteria metabolism that we've already studied. But then there are some elements that are very unique to archaea, and that's why it has been put away from the bacteria as a special group now, as a domain now. They have unique ribosomal RNA gene structures, and some of them are capable of methanogenesis, which is an important property only found in the archaea. Also, in terms of morphology, physiology, reproduction, and ecology, they are very diverse. Within the archaea group, they are very diverse also. They are known for their ability to survive in anaerobic conditions, hypersaline conditions, pH extremes and high temperature habitats. So usually, up to now, we haven't found any archaea that lives around us or on and be capable of causing any disease or infection in us. So they are definitely the ones that live away from us, not in our everyday surroundings, but in very extreme um, environments as such. Also, they may be found in the marine Arctic temperatures and the tropical waters, very extreme cold and extreme hot areas also. These shape size of these archaea, they almost look like bacteria, and they may be cocci rods. These are the common shapes, but there are other shapes that will also exist in different ones. But up to now, we haven't looked at any spiral or yeast-like forms in the archaea. Well, we haven't discovered everything yet. We may find them. They may have branched or flat shapes also. Their sizes are almost like your bacterial sizes, typically between 1 to 2 times 1 to 5 micrometers if they are rods, and the cocci diameters could be 1 to 5 micrometers also. The smallest one, as we know, have been observed about zero point 0.2, which is also the size of our mycoplasma also. And some of the archaea are very large, and they may reach up to 30 millimeters also in length. So here are some of the tables that give you some comparisons and common features between bacteria and archaea. I won't run through all of them. I just want you to understand the terminology to get this terminology set as to what are some of the features, structures that you see in bacteria and archaea. You familiar with the word plasma membrane? Yes, we also call it the cytoplasmic membrane or plasma membrane. Plasma membrane is supposed to be the structure that should be present in all cellular forms of life. And that is the membrane that covers the cytoplasm. And so plasma membrane is a requirement for all living cells. And this plasma membrane has the function of being selectively permeable. It has, in bacteria, it is an important one where metabolic processes are also located, such as respiration. Even photosynthesis is located in the plasma membrane. And of course, if there is chemotaxis in the bacteria, these cues or to be able to respond to chemicals or even surrounding all the function is located in the plasma membrane. So very important part of a cell there. Some of the microbes that survive in the aquatic environments, they have gas vacuoles that allows them to be buoyant and come to the surface and survive there. Ribosomes, you're familiar with that word, required for protein synthesis. Ribosomes of bacteria and archaea are just the same. They are 70S ribosomes, and we know that in your eukaryotic ribosomes are considered to be the 80S ones. There will be some inclusions in the cells of bacteria and archaea, which will be for storage of carbon, phosphate, and other substances. Now, a very important term, which where all of you make a mistake, is that bacteria and archaea do not have a nucleus. So we never call it a nucleus. We have coined a term called nucleoid now, okay? So nucleoid is the area where the genetic material is kind of located, but it is not covered with a membrane. So please remember, bacteria and archaea have nucleoid, not a true nucleus. There is an important part in the structure of the cell, which is considered to be periplasmic space. And this periplasmic space is very different in your gram-negative versus your gram-positive bacteria. Gram-negatives having two membranes, cytoplasmic membrane. Gram-negative have cytoplasmic membrane and an outer membrane. So the area in between the two membranes is a contained area, which we will call periplasmic space. And this, of course, is like a storage area for that bacterium, and it will contain a lot of proteins in there, and a lot of processing and important transfers will occur through the periplasm. So if you're thinking of a gram-negative bacterium, there is a very thin peptidoglycan that is within the periplasmic space. Whilst in your gram-positive bacteria, since there's no outer membrane, the space between the cytoplasmic membrane and the peptidoglycan is considered by some authors to be the periplasmic space, and these will be either very small or they may be absent also. So gram positives have do not have this advantage of having an enclosed periplasmic space. So whatever is in the periplasmic area in the gram positive bacteria have to be somehow hooked up or attached to the peptidoglycan. Otherwise, that will float away as an exotoxin or an exoenzyme or whatever it is, right? Cell wall is a very important component of bacterial and archaeal cells. They provide shape and protection. And of course, the differences in the cell wall of archaea and bacteria are considerable. Even though they both have cell walls, we know bacterial cell walls have peptidoglycan, which is very special to them only, whilst your archaea will have some cell wall, some components like peptidoglycan, but this is not the true peptidoglycan that we will study today, also later when we talk about the cell wall structure. So there is some difference and some similarity there. There are capsules and slime layers in bacteria and archaea. In archaea, capsules and slime layers are rare, but they may be found on some of them. But this is a very important property or a structure for bacteria, which helps them resist phagocytosis, because these bacteria are capable of causing diseases, and they have to get into the host, and the host has phagocytic cells. So capsules and slime layers definitely help in warding off this phagocytosis. Also, slime layers are very well known to allow adherence to surfaces where they have to stick to the tissue when they are causing the disease, or when they are in an aquatic environment, they need to stick onto the rocks or stuff like that. And then we are familiar with the term fimbri and pili, or these are also meant for attachment. And we know even though structurally they are very similar, pilus may be the long one, fimbri may be the short ones. And pilus are the ones that we definitely use that term only for appendages that allow conjugation and even transformation, whilst your fimbri will be the one that will, and also some pillars, which will allow a special kind of motility that we will study, is twitching and the gliding motility. So rather than the swimming motility that comes with the flagella, pili and fimbri are the ones that will be allowing the bacteria to have twitching or a gliding motility. And finally, of course, we have endospores, and this is found only in bacteria. Archaea, we have not found any archaea to have endospores, or bacteria endowed with this property where they can survive harsh environmental conditions when they go into the endospore form. So just to give you a brief summary of what you already know and warm you up to the topics as we go along. Here are some of the differences that you will see in the bacteria and the archaea, and some similarities from another book. I give you a lot of these information charts so that you can always refer to them when you are studying or presenting a paper or doing anything else. So we'll just run through these. Here is a third group also present in this comparative chart, where we have the bacteria, archaea, and eukarya, all of them, and very specific kind of information is here that we will work on as we go along when we talk about each of these components of the cell, or the property of that cell, how different they are in each one of them. So these were all for references for you.